Good morning, everyone. Thank you for joining this webinar. So we are going to be having this webinar titled Revolutioning Lab Automation Across Sub-Saharan Africa with SPTLabtech and LASEC. My name is Alannah Theron, and I am the Biotechnology Application Specialist at LASEC. I'll be your main point of contact for all things related to SPTLabtech, providing both technical and application support. This partnership is a collaboration designed to expand access to cutting-edge laboratory automation across the entire Sub-Saharan Africa. In this session, we will explore how SPTLabtech's innovative automation technologies, combined with LASEC's regional expertise and support, will empower scientists, researchers, and diagnostic labs with more efficient, reliable, and accessible solutions. The key takeaways for this webinar include how SPTLabtech's automated liquid handling solutions enhance efficiency and accuracy, how LASEC supports local access, implementation, and ongoing technical expertise. We will have a Q&A session with our team as well to address all your specific needs. So if you have any questions throughout the entire presentation, please feel free to put your message into the chat box. For those who may not be familiar with us, LASEC is a leading supplier of scientific and laboratory equipment across Africa. With over 75 years of experience, we partner with globally trusted brands to provide tailored solutions, technical expertise, and a dedicated support across a range of industries, from clinical diagnostics and research to education, as well as industry. At LASEC, we pride ourselves on not just supplying products, but enabling science through strong partnerships, training, and service excellence throughout the African continent. I will be your main point of contact, as I mentioned previously, at LASEC for any requests or technical queries related to SPTLabtech. So please don't hesitate if you would like to reach out via email for any questions or any information that you would need. We're also fortunate to be supported by Alannah Bamford, the distribution sales manager who works very closely with us through SPTLabtech. Without further ado, it is my pleasure to introduce Paul Lomax, the head of genomics at SPTLabtech, who will be also presenting today's talk. Since joining SPTLabtech in 2015, Paul has been responsible for driving innovation in laboratory automation by working closely with researchers and collaborators to develop new instruments and applications that address the evolving market needs in drug discovery and genomics. This has included the mosquito and dragonfly instruments that utilize novel, patented, positive displacement pipetting and non-contact dispensing technologies. More recently, he led the development of the Firefly, an all-in-one liquid handling platform, guiding the project from initial concept all the way to launch. With over 20 years in experience in automating sample processing, Paul's expertise spans academic, clinical, environmental, and biotech sectors. So without further ado, Paul, I'm handing over to you.
Morning, everybody, and thanks to LASEC for inviting me to speak this morning. So as Alannah said, I'm going to be talking mostly about our liquid handling platforms. But before I do that, I will give you a little introduction to SPTLabtech and just a broader view of the range of products that we do. Alannah, just check that you can see my slides okay?
All good.
Perfect, thank you. SPTLabtech is a global organisation. Our headquarters are in the UK, where I am now, and quite a bit of our manufacturing is done in the UK, but we also have manufacturing sites in West Coast US, also in China, and then we have a number of, we also have a site in Germany, and then we have a number of commercial hubs across the globe. In the US, in the UK, in India, China, and Japan. And we are directly selling to the US and Europe and quite a bit of Asia as well. And then we work through a network of distributors for the rest of the world. So, and we're very selective with our distributors. You know, we look for distributors that will really support our customers and work collaboratively with us. So we're delighted to work with LASEC, who absolutely kind of fit that mold. We also have a diverse and large customer base, and we are working across life sciences. So we work with a lot of the major pharma and biotech companies across the globe, a lot of the major academic facilities and also government facilities, as well as other areas such as contract research organisations and agrochemicals. So it's really quite a diverse set of customers and a fairly broad set of applications that we cater for as well. In terms of kind of what our focus is, we really focus on sample preparation, and there's three broad areas that we work in. We've been established now for, we established back in the early 2000s, so we're heading for something around 25 years old now. And our kind of roots are in liquid handling. Our first liquid handling product was the Mosquito, which I'll talk a little bit more about later, but also products within sample management. So I'm going to talk mostly in this presentation about our liquid handling range, but just to also let you know at this point that we also do a range of sample management products, which are essentially focused on the storage of 2D barcoded tubes and rapid access to those stored tubes. So things like small molecule storage at room temperature or -20, large molecule storage such as antibodies, plasma, other biological samples, DNA, RNA, and so on, typically at -80. And we also do a range of tube handling equipment as well. So tube pickers, sorters, and so on for the organisation of tubes ready for use in applications. So that's the sample management area. And then we also have a division which focuses on sample preparation for cryo-electron microscopy, which is the transfer of very small amounts of protein onto the grids that are used within cryo-electron microscopes. So kind of very specialized area, but you'll see when I come into the liquid handling presentation that it links with some of our legacy in one of the kind of application areas that we deal in for liquid handling. So I am going to focus the rest of this talk now on the range of liquid handling products and technologies that we supply. So the liquid handling range is broadly split into four areas. So we have the apricot range of products, and this was through acquisition several years ago. Apricot was an existing brand in the market, had been around for around about 30 years. So a lot of experience and pedigree in the market, providing multi-channel pipetting heads predominantly, and actually you'll find those apricot technologies across a wide range of brands. You won't know it because they're sold through OEM to quite a number of the leading liquid handling companies across the globe. So, you know, some of our competitors even are working with apricot technology within their systems. So a really well tried and tested, proven technology based around air displacement pipetting. Then we have the Dragonfly range, which is a non-contact dispensing system. And again, I'll go into more detail on that. There's a few different variations on that system for different application areas. And then the Mosquito, which, as I mentioned earlier, was kind of the first venture into liquid handling for SPTLabtech a good while ago now, and it's evolved over time and found its way into many new application areas. And then most recently, we've added the Firefly. About 4 years ago now that we launched the Firefly, and then more recently the kind of shelving unit that you see to the left of the Firefly, where the new sticker is there, is the Firefly Plus, which is an accessory product for the Firefly that's been added recently, and I'll go into details of what that does and so on. So I'm going to go through these areas kind of one by one just to give you a little bit more detail. And there'll be opportunity for questions at the end of the presentation. We also work across a broad range of applications, but I think you can probably, the vast majority of things that we work in kind of fall into one of these three categories. So we do a lot of work in genomics, particularly with Firefly, Mosquito, and Dragonfly. So things like PCR and qPCR setup, library prep, a lot of single cell applications, transcriptomics, surveillance, metagenomics, and a whole array of genomics applications, both at standard volumes, kind of replacing manual processes with automation, and also with a focus on miniaturization with some of our products and focus very much on cost saving and so on. Then where we kind of started out originally with the mosquito was more focused on drug discovery, but that's still a strong element of what we do. And things like assay development, assay setup, medicinal chemistry applications, proteomics, and more genomics that we're seeing within drug discovery now as well. And then finally, a kind of more specialist niche area, but we kind of very much market leaders in this side of things, which is in structural biology. So I mentioned the cryo electron microscopy, which have been kind of the new thing in structural biology over the last few years, but we've got a long history in structural biology in the protein crystallography methods. So we've got special variations of mosquito that are designed for setting up protein crystallography applications, things like sitting drop, hanging drop, liquid cubic phase, and so on. And we are very much a kind of a go-to solution for that for many crystallographers. So I'm going to talk a little bit in detail about the apricot now, the apricot pipetters. So there are three broad categories here. So we've got the S series, which is certainly, I think, amongst the most popular products within the apricot range. We've also got the personal pipetta, and we've got the DC1. So focusing a little on the S series, and these are really, I think these are often like the first step into automation for customers that have previously been working with manual liquid handling. So a typical customer for this would be someone who's spending a lot of time at the bench pipetting, using multi-channels, maybe working with full 96 well plates or full 384 well plates, and finding it challenging to actually keep up with the level of throughput required, or perhaps struggling with the consistency, particularly the multi-user environment, and really looking for a step up to kind of reduce the pain of, well, can be the literal pain of pipetting and things like RSI. But also looking to improve data and consistency with, taking that human element out of a process. So the S series, they have a series of user exchangeable cores, and I'll expand on that with a video that I've got that I'll show you in a moment. But essentially, it makes it a very flexible system where you can actually exchange the volume range. You can exchange the format, so you could have a 96 pipetting head or a 384 pipetting head, and you're also able to work with a wide range of plate types, even down to kind of lower density plates like 24 well plates, but predominantly people are using these for 96 well plates and 384 well plates. You can also work in a column-based approach, so you can load single strips of tips or single rows of tips, and this will allow you to do things like serial dilutions or work down a plate row by row. So it is quite flexible, but they're typically used for kind of limited numbers of steps, so kind of single step operations where you're doing a transfer from one plate to another and so on, and really replacing, giving you a higher throughput, more reproducible approach than, say, a multi-channel pipette. And there are two types you can see in the image. There's the S1, which has a single stage, so you're manually moving plates in and out of that. And then there's also the S3, which is the one on the right here, which has got three positions, so you can put like a source plate, a reagent plate, and a destination plate in there. That just gives you a little bit more more automation. Then we've got the personal pipetter, which is a slightly more multi-step automation system. So it's got 5 deck positions, so you can kind of program now more steps within this, and it can perform more of an application. It doesn't have the exchangeable cores, so you typically choose which format that you want, and there's an array of these. Again, it can cover from 24 well plates through to 384, and actually this one is a higher precision machine, so it can actually extend to 1536 plates as well. So if you're interested in that area, this could be a good option for you and give you the ability to go into 1536. Again, it can do both the column-based pipetting, so it can do things like serial dilutions with a single column of 8 tips for 96 or 16 for 384, and it can also work in rows, again, like the S series. And then finally, the DC1 is what we call a 4-in-1 head. So this is actually not a full array of tips. So it's not a full array of 96 or 384 tips. It works in a column, much like a multi-channel pipetter, but it can actually, within that column, you can work in the row format. So it is actually a 12 tip or 24 tip pipetting head. It can pick up either columns or rows of tips, and it can also pick up individual tips as well. So it gives you quite a lot of versatility. Imagine the sort of flexibility you've got with a single channel pipette and a multi-channel pipette all rolled into one. And then it's also got a larger deck area of 8 positions, so you can actually place a number of different bits of labware, tubes, and so on. So this is a kind of more versatile, albeit lower throughput workstation, but it's really designed at kind of simple applications that you can replace with a automated approach rather than a manual approach. And again, giving you the benefits of more consistent pipetting and so on. So this is all of these are really designed to be out of the box automation, meaning that pretty much they can be placed on the bench, and with a very small amount of training, you can be up and running with them very quickly. And they're multi-use instruments that anybody in the lab can walk up to and easily use, much in the way that anybody could pick up a multi-channel pipette and use it. But really driving improved data quality versus manual pipetting, and reducing the kind of the physical strains of pipetting, particularly around RSI and so on. So I'll just end this section with a short video sequence of the S series. So you can see here the unit's very small and compact, so it doesn't take up much bench space. You can see the moving deck here. Here it's just showing a simple transfer from one 96 well plate to another 96 well plate. It uses air displacement technology. So the same sort of technology you've got in most pipetters, and you can see there how the core is removed, so the block can simply be lifted out, replaced with a different format block by the user. So it's very versatile, and it's got this simple elastomer gasket seal that seals between the tips and the pistons. So the tips can actually be reused if that's applicable to your application. There's no kind of destructive seal on the tips. And it really is an easy to use system. So firstly, load the tips simply in this tray, press a button, it clamps the tips. You've then got a very easy user interface where you select the type of plates that you're using, different labwares. You select the volumes that you want to do in the operation. And it's a very simple, clear screen that's used, and hence the kind of limited kind of training time required. And then once you've selected your method, you run your method and you're done. The cores also give you a very versatile configuration, so from small 96 cores through to 384 cores, and a range of volumes from 0.5 microliter up to 1 milliliter. And then you've got a range of liquid handling operations that are covered within the software, just simple things like copy, reformat, and so on. So hopefully that gives you a little flavour of the apricot range. I'm now going to move on to the Dragonfly. So the Dragonfly, so whereas the apricot range is very much individual pipetting, well to well, and moving from plate to plate and working within plates just as you would with a multi-channel pipette, the Dragonfly is actually a dispenser, so a one-to-many dispenser. So on the left here, you can see there are a number of dispensing syringes. There are different variants of the machine, so you can have 3, 6, or 10 of these dispensing channels. The picture here shows a 10 channel dispenser, so you've got two banks of 5. Each of these syringes is capable of dispensing from 200 nanoliters up to 4 milliliters. So there's a 4 milliliter capacity in the syringe, and these syringes are fully disposable. They use a positive displacement technology, which I'll talk about a little bit more. And again, there's a video that will explain this a little bit better. But you're actually using a syringe where a plastic syringe barrel comes into contact with the liquid and forces it out, meaning it is really liquid class agnostic. So we often say we can dispense easily with the same settings from 70% ethanol through to 70% glycerol, and you don't need to change anything. You just put the volume in and it will work. It can be used for simple reagent dispensing. So you could have each syringe set up with a different reagent and then just use it to dispense out to plates. You can have the same reagent across multiple syringes if you want to go faster. Obviously, each syringe is then only dispensing to a part of the plate, which increases the throughput. But you can also use it in quite sophisticated ways as well. So you can be delivering different reagents to different wells at different volumes. And this really, for things like assay development, where you might want to be trying different volumes of reagents, different wells, and maybe using design of experiment tools, things like JUMP or Synthase, which can come up with rather quite sophisticated reagent maps. You can just export those sorts of files into the software and then run those complex methods. And that's really something you just can't do manually. It's just too complicated to be winding volumes up and down pipettes across multiple reagents. So it opens up a whole new world of possibilities when it comes to things like design of experiment. It also can work across a number of different plate formats. So typically 96-84 and 50-36. It can also work with lower density formats as well, so like 24 and 48. And the sort of common applications that we see with Dragonfly, a lot of genomics applications, so things like qPCR, PCR setup for enzyme dispensing, also things like normalization, because we can dispense different volumes to different wells from a CSV file. I've mentioned assay development, but also dispensing kind of things that are typically thought to be more difficult to dispense, cell suspensions, hydrogels, beads, magnetic beads, and so on. So it's a really, really versatile dispenser where you're trying to broadcast across a whole plate. We also do have a specialized version of the Dragonfly, which is Dragonfly Crystal, which is designed for rapid and accurate preparation of complex crystallization gradients and optimization screens. So it's a slightly different variation with specific software, but that's really geared towards people working within protein crystallography to assist them, again, kind of making what can be challenging experiments really, really simple and reliable. So again, I'll finish this section with a short video just to give you a better flavor for the Dragonfly discovery. So again, the unit's very compact, small amount of bench space required. It's got a single stage position where you place the labware. Inside the machine, the dispense heads are driven by individually. So each of these positive displacement syringes is driven by an independent motor, which can deliver volumes as low as 200 nanoliters. And these are aspirated from a range of different reagent reservoir types, from low dead volume to higher capacity and so on. And a wide range of compatible labwares are possible. It's a simple walk up and use instrument. Generally, it's one of those things that anyone in the lab can walk up to and easily use the software. And at the heart of it is this positive displacement technology. All the liquid contact parts are disposable, but this positive displacement is what's giving the reproducibility and accuracy down to these very low volumes, as I mentioned, like down to 200 nanoliters, and also giving it the ability to cope with a wide range of viscosities, volatilities, particulates, things like cells and beads. Here you see it dispensing with two different reagents simultaneously in a 1536 well plate. Here, just dispensing a single reagent into a PCR plate. And there are also some accessories that help with high throughput options. So it's got an API, it can be connected into an automated work cell, and you can have a high volume reagent supply through the valves that we show here and the peristaltic pump. So you can have a large reservoir, bottle of liquid, and you can have a number of these autofill reservoirs, as they're called, that can be daisy chained together as well. So you can have multiple syringes being fed by the same reagent if you want, or just a single or a double. So it's very flexible, and you can also mix and match those AFRs with the manual reservoirs. So you've got a lot of flexibility on the configuration. So I'll now move on to the next section, which is the mosquito. So with the mosquito, we're now actually back to the pipetting system again. And there's a number of different versions of the Mosquito, again, designed for our specific application areas. Now, this is probably the closest thing that we supply to a multi-channel pipette, because these systems work in a single column of tips. But interestingly, the tips are supplied. You see that the disc feature at the top of the system, that's actually a spool of disposable tips. So there's up to 36,000 tips on a single spool that can be loaded on. So typically the system can be loaded with tips, and for some customers that it may be weeks or months before that needs to be replaced. It cuts down an awful lot on plastic wear because you don't have racks for the tips. And from an environmental point of view, it's really, really great in terms of that. And also from a space point of view, it's great because you don't have to worry about shelving and so on to store the tips. But the key thing about the key difference between this and say a multi-channel pipette is the volume range. So these are really designed at miniaturization, and I'll go through the details in a little more shortly. But essentially, we can go down as low as 25 nanoliters with these systems, up to 5 microliters with 2 versions of the machine. So with the HV is the high volume, the LV is the low volume. We have a range of systems focused on genomics. So we have the HV, the LV, and recently we've added what we call the Gen 3. The HV is in bold there because it's the most common one that we sell for genomics applications. So this is a system that works. It has 5 deck positions, the HV, so you can put up to 5 bits of labware. So again, things like a source, a destination plate, a magnetic block position, maybe a cooling position, and so on. And will allow you to do it's semi-automation approach, but it is pretty full workflows, and we've got a wide range of methods for the Mosquito genomics. We've got over 40 verified methods for different library preps and so on. So typically customers are using this for miniaturized versions of library prep, qPCR, PCR setup, quantification, normalization are very typical applications, but very much focused on miniaturization. So where customers are looking to reduce the volumes, reduce cost, and increase the throughput. It's been particularly used in a lot of single cell applications where people wanted to do plate-based single cell applications where the costs can be very high. And this has made kind of experiments affordable by reducing the volumes by as much as tenfold or even more. So that's the genomics area. Then crystallography is kind of been one of the real areas that we've been successful with Mosquito for many years now. And again, there are specific products. There's the Crystal 3, which is the most popular product right now. But we also have the LCP, which stands for liquid cubic phase, which is kind of very specific for that application. And then we also have a general LV instrument, and that's generally where people want to do protein crystallography, but maybe other applications as well. It gives them a bit more flexibility. But typically this is used for protein crystallization screening, so things like drop setup and liquid cubic phase. And then finally, the drug discovery element, again, this is one of the areas that we started with the Mosquito. So there's a range of both the low volume from the 25 nanoliter volume up to the HV, which is up to the 5 microliters. And then there's also a version called the X1, which is just a single tip for things like cherry picking and so on. So the sorts of applications that are typical here, assay ready plate preparation, serial dilutions, cherry picking, and so on. And we find that often you'll find these systems often standing next to things like acoustic dispensers, where this kind of bridges the gap. It offers a more rapid solution to that range that kind of falls between traditional liquid handlers, which are typically being used from 2 microliters and up, to things like the LabSite Echo, which are typically up to the kind of small nanoliter scale. So this really bridges that gap and is a really useful tool for that area. So again, I'll talk a little bit about the core technology, and then again, I'm going to show you a video just to kind of wrap this section up. So core to the Mosquito is the pipetting tip itself. So on the right hand side, the very far right, you see a tip layout there, and it's essentially got a polyester backing tape So that's what runs through the spool and gets loaded into the system that you'll see in the video in a moment. It's got a stainless steel piston, and it's got a high density polyethylene barrel. So again, the liquid is being forced out by the piston. And that's what gives us the ability to have, you're not relying on an air cushion as with a normal pipette, which allows us to go to these very small volumes. So we do two sizes of tips, and they work in two different systems. So the LV uses the small tip, which works between 25 nanolitres and 1.2 microlitres. The HV works with a larger tip, which goes from 500 nanolitres to 5 microlitres. And this is true positive displacement, and it is actually a piston forcing the liquid out. Those tips are available, they're disposable, they come on those reels, the spools that I mentioned, and we do have the option of gamma irradiated tips as well for sterile applications. So they work across a wide range of liquid types, low and high viscosity, that kind of 70% ethanol to 70% glycerol kind of comes in again, but also things like particulates, beads, and so on. And there's a range of functions within the software, so simple copy, transfer from one plate to another, things like multi-aspirate, multi-dispense, and mixing, and you'll see all this in the video that I'll show you in a moment. They also have really negligible dead volume, so you can really kind of cut down on waste of within reagents and so on, particularly where they might be expensive enzymatic reagents in things like library prep. You don't see bubbles, you don't see clogging, you don't see dripping. So these are really reliable, robust pipetting systems. And they're actually very gentle on complex molecules. So large molecules like DNA, enzymes, proteins don't get sheared by these tips. They're very gentle in the way that they operate with them. So it's a great fit with the application areas that we're working with. And I think I already mentioned that there's a lot of tips on a single spool, so you can load the system up. And it could be, running for weeks or even months, depending on the throughput levels before the tip spools are needing to be replaced. So I'll finish this section with a short video as well. And here you see, so 16 tips. It can work in 8 or 16 mode. So it's just aspirated from initial reagent. It's now going to move over to picking a different diluent, and it's first laying down 100% of that diluent, and then it's going to go through a series of serial dilution series. So you can see it mixing, and then it's going to aspirate back from that what it's just dispensed, and move to the next, and build this seal dilution across. Here we're just doing this on a plate lid, so you can actually see what's going on. Obviously, normally this would be going on within in this case, within a 384 well plate, but it could also be within a 96 well plate, or it could be within 1536 well plate. In the case of 96 well plates, we can load just 8 tips. So there's a different spool of tips for that, with the pitch space in between sets for a 96 well plate. And for 1536 well plate, we can actually quadrant, so we can work one quadrant at a time through the plate. So it's a very versatile system, but one of the real benefits is that the ease of use of this, you don't need to worry about things like liquid class setup, like air gaps, speeds, and so on. You can generally just set the volume and run, because the positive displacement approach will work with any liquid, regardless of the viscosity at the volume that you choose. So I'll conclude the mosquito section there, and then finally move on to our kind of latest product release, which is the Firefly, and then more recently the Firefly Plus. So I mentioned the Firefly was launched about 4 years ago now, and in terms of technology, it is combining kind of 2 tried and tested technologies in terms of pipetting. So it incorporates the Dragonfly dispensing technology, and it also incorporates the apricot pipetting technology. So it really was kind of taking tried tested liquid handling technology and repackaging into a new system that gave it a lot more flexibility and the ability to do end-to-end applications. And then we also more recently added the Firefly Plus, which actually enhances the capacity and provides greater walk-away capabilities. So I'm going to go into both of these in a little bit more detail. The really nice thing is the Firefly Plus can be added to any existing Firefly, so it really is a modular system. And customers like that that ability to maybe start with the Firefly on its own system on the left, with a view that, you know, should their walkaway needs or capacity increase in time, they can add the Plus later. Obviously, some customers come straight away knowing that they've got the greater capacity needs and kind of go for the full Plus system from the start. But that modularity gives a lot of future-proofing possibilities. So I've got an image of the Firefly Plus here, but I'm going to talk first on the right-hand side about the features of the Firefly itself. You can see there is some movement in this image, which kind of gives a flavour of it. But again, I've got a video which will go into a lot more detail shortly. So there is a touch screen. Everything is driven from that, so that the unit itself doesn't need any more space. Again, it's very compact. So you can see that the whole Plus system is only just over 1 meter wide. A Firefly on its own is just over 600 millimeters wide. So it's a very small amount of space on the bench. The way we do this is by using vertical space. So you can see on the right hand side of the instrument, we've got the dispense heads, and you can have 3 or 6 of those in a Firefly. Essentially, this is the same positive displacement non-contact technology that we've got in the Dragonfly. So this is designed for going from a single reagent to many wells across a 96 or 384 well plate. Then on the left hand side, we have the same pipetting technology that we have in the Apricot S series systems and P series systems, and this is actually a 384 well pipetting head, but it can be used in either 384 or 96 format. So we supply tips in both formats. It also incorporates a gripper that you can see in the kind of little GIF loop here, which is can drop down below the head and pick up and move a plate from different positions on the decks. You've also got moving decks, and you can see again in the image here that the top deck is moving left and right. This is how we get the movement between the dispense head and the pipetting head. With the lower deck will also move across in the same way, and you can use, there are 8 positions on each of the decks, so you can use you can load up plates, tips, other bits of labware, magnetic blocks, and a whole array of plate-based or plate formatted bits of labware. Typically, we use the lower deck for tips and the upper deck for labware, but you can put labware on the lower decks as well. There's also below deck additional parts as well. So below these 2 moving decks on the right hand side, there's an incubated position for reagents, so we can heat or cool reagents that can be accessed by the dispense heads to aspirate. And then on the left hand side, underneath the deck, there is an incubated plate position. So again, we can heat and cool plates. And there's also a shaker position. So the gripper can move plates between the layers and down into those that lower section to incubate and shake plates. Then on the left-hand side, if you're running along, like a library prep application, for example, you will exceed that 16 deck capacity that we've got on the machine, and you'll need extra tips, certain things like extra sealing lids and so on, and possibly extra plates to run the process end to end. So here we get the extra capacity through the shelving system. There's a small robotic arm in the Plus as well, which can move things in and out of the Firefly Plus. And then the final thing that's in there is the on-deck thermocycler. So you can't see it in the image, but sat at the bottom of this unit is an automated PCR machine. So again, we can move, once a plate has been prepared, we can move the plate into the thermocycler, add a lid to it, and then run the process, and then bring the plate back out to continue. So it really kind of gives the opportunity to run kind of full end-to-end workflows, particularly in genomics applications, but not exclusively. There are other kind of drug discovery applications as well that kind of benefit from this extra capacity and the options to incubate with a thermocycler. And so I'm going to give you just more of a view of the Firefly in action through this video to kind of wrap up the presentation today. So you can see, first of all, that the software, again, is very intuitive. Obviously, this is a system that's doing a lot more, but again, it's still very easy to use and very accessible to people who even, haven't got a lot of experience with automation. For a protocol to be run, it gives a full list of the required consumables, and it has a really nice loading guide. So if you've got people in the lab that might be a little bit afraid of using automation, it really can be set up in a way that really helps them and guides them through. So when they come to the system, if you want them to run a protocol that's been set up within the lab, they can just be guided through all the steps they need to do. So things like loading the syringes, it'll bring the heads to the front of the machine so they can actually load the syringes into place. It will show them exactly where to load each bit of labware. Within the programming, it can be, all of these things can be named specifically, so you can give things, you can even change the colors of things to make it easier to kind of follow. So you can really kind of personalize how this setup is done, and this loading guide makes it really kind of as foolproof as it can be to make sure that everything is loaded correctly into the system before use. And the system also, as you go through this, will kind of move the decks automatically to the most convenient position to load things on. So you can see a magnetic block being loaded into position there. We can use a lot of the standard commercial magnetic blocks that you may be using in the lab already, things like alpaca, permagen, and so on. So it's a really good way of actually just making sure, I think, and avoiding the sorts of user error that that can occur sometimes with using automation. And then the same goes for actually loading the reservoirs with reagents and so on. So it really takes you nicely step by step through this process. And once you get to a point where the setup is complete, there's a final screen just to check that everything's in the right place. So this video actually just shows elements of a bead cleanup, just some of the kind of key steps that you'd see within that. So the first thing it's going to do is load a set of 96 tips on, so it can seamlessly load different tip formats and different tip volumes. So there's 2 volumes of tips. There's a maximum 50 microliter and a maximum 125 microliter tips, and they're available in both 96 and 384 format. Here it's loaded on a set of 96 tips. And the first thing it's going to do, having loaded those tips, is actually aspirate the reagent. So here we're just using coloured liquids, but this essentially would be a bead dispense. So I think it's using two syringes for this dispense. So you can see the 2 leftmost syringes are used. And that's generally just a trade-off between speed and dead volume. Obviously, you've got, for each extra syringe you use, you've got extra dead volume from an additional reagent trough. So here it's actually dispensing across this 96 well plate. So each syringe is doing half the plate, so 48 wells each. And as you can see, it does this pretty quickly. And it works very, very well with a wide range of magnetic bead types. Having done that, it's now going in with the, it's going to transfer the sample across into those beads. So the pipetting head is going into the source plate. Again, in this case, just another coloured liquid in this demo. And now it's going in to actually mix those. And you've got full control over things like the mix speeds and so on, to make sure that you get efficient mixing within the system. The nice thing is we've got a lot of protocols across a wide range of vendors for things like library prep, where we've already got pre-written and validated methods, and these are available on our cloud system. So you can actually download them directly to the machine, or you can download them to your laptop, for example, and then transfer them across your network to the machine. So there's very easy ways to get access to a wide range of applications that are ready to go. So having transferred and mixed the samples, it's now picking up the plate and transferring it to the magnetic block. You can see the gripper now comes into play here, checks that the plate is there, and then moves it into position on the magnetic block. And then another nice thing that you'll see in a moment, I think you can just about make it out on this video, is the magnetic blocks are typically sprung. So when you're actually going through wash steps where once you've pulled the beads down, you actually want to then remove everything else from in that well, you can actually use the spring feature to depress the magnet. We typically are aspirating actually below the effective well bottom by forcing the spring down slightly, which means that we can actually aspirate everything in the well plus a little bit more, and you'll see this trailing air gap that you see, just ensuring that everything is taken out of that plate. And then it's now just transferring to a waste plate. It's just doing a slow dispense to the waste plate just to make sure everything is removed from those tips. So it's as well as being very easy to use, it still has got a lot of flexibility in terms of the control that a user has, and the ability to fine tune this. So certainly, you know, people with experience in automation who want to do those sorts of fine tuning steps can still do that, as well as it being very accessible to users. So I'm going to stop it there. Obviously, there's an awful lot more I could tell you about the system, but the main kind of things about this I would stress is that, you know, we found that the Firefly has been, I think, one of the main things that's come across is the ease of use of the system, as well as the flexibility and the power that you've got. You've kind of got two liquid handlers in one. You've got the pipetting and the non-contact dispensing. It's pretty unique in the market in that respect. And in terms of, you know, lab space these days is often very constrained. So just how compact the system is and using the vertical space how it does has really been important, because there are some labs that simply don't have the space for a big automated system, whereas they can fit a Firefly in and it can provide a lot of power for them. So I will end it there, and just kind of finish on the slide, just again reminding you of what I've talked through in this presentation. But we've got the kind of entry-level systems with Apricot. So if you're looking kind of for the first step into automation, just taking some of the strain off, manual pipetting, increasing throughputs and looking for better consistency and accuracy in your pipetting, then the Apricot range is a really great place to start. If you're looking for a robust dispensing system, then the Dragonfly is an excellent choice and can work with a wide range of reagents. And, you know, both of those systems, Apricot and Dragonfly, are very, very kind of simple systems to use for a wide number of users across the lab. And then you've got, you know, if you're looking to miniaturize or perhaps specialize in something like protein crystallography applications or miniaturized genomics applications, then the Mosquito is an excellent choice for that. And again, very easy to use and set up. And then finally, if you're looking for a kind of more advanced automated system with kind of larger walkaway workflows, then that's really where the Firefly comes into its own. And again, it kind of continues that theme of ease of use and flexibility. So I'll end there, and yeah, thank you for your attention.
Yes, thank you so much, Paul. It was a very informative presentation. I think one of the big benefits of SPTLabtech is the wide variety of automated liquid handling solutions that you have. Thank you everyone for joining, and have a lovely day. Thank you, Paul.
Thank you. Thanks, everyone.